Excellent. So hello to all the new students. We have been doing a Cambridge IGCSE at GCSE, but any sort of on any of the GCSEs will lead well into the AQA A-level course. And once you've started, we'll be going through all the content. So as long as you've got a bit of a baseline and you are fairly proficient at maths, then you should be fine on the course. I'm just going to get up the PowerPoint today and share my screen, so bear with me.
I'll leave you to it, Mrs. Richardson. I might pop back in later, but that's superb. Well done, everyone. I'm pleased to see there's been a good uptake. And I'll speak to you soon. Cheers.
Yeah, bye-bye. Right, bear with me one moment. We're just going to look at a little introduction to the course. Okay, can we see my screen now? Here we go. So, why study biology? Biology is, obviously I'm biased, but it's a massively interesting subject. It lets you understand how your body is functioning, how we got here, how we can impact the environment, the interactions between us and the environment, the skill set that you need to do biology, because it's such a broad subject. There are also many skills. So you need good literacy skills. You need to be able to read and articulate your thoughts very clearly and precisely using key terms. You need to use those accurately. Numeracy, you need to be fairly adept at numeracy. Now, I did not take A-level biology. I did quite well at GCSE. Really, the maths in A-level biology is no harder than about an 8 at GCSE, a grade 8 at GCSE. So it shouldn't be any harder than that. We're not going into sort of A-level maths, but you need to be able to apply it in the right places. Invest 10% of all the marks on all of the exam papers will be maths, okay, maths questions. 15% of them will be investigation skills, so the nature of science, so hypotheses, variables, controls, means, reliability, precision. Then we also bring in ethics as well. So there's quite a lot of 15% of the marks are the investigation skills. Biology is always topical, and we're going to look at zoonosis in the second part of today's session, which obviously is very relevant to what is going on at the moment. Obviously, I am biased, but I believe the teaching and learning in our biology department is one of the best in the school. We have 2 A-level examiners within the department, over 45 years worth of experience teaching in the department. We have a range of activities, lots of independent learning, creative tasks, self-led, teacher-led. There's lots of support in terms of drop-in sessions, but also teacher-led revision sessions in year 13. So there's lots that you can be, lots of support in the department. So we'll just have a look at a few of these quotes. Nothing in biology makes sense except in the light of evolution. Pretty much everything you can apply to evolution. And evolution links with things like geology, chemistry, history, quite heavily. Our ultimate goal is to use biology to improve outcomes. A lot of people want to know how they can then apply their knowledge. And with biology, it's about finding out the way that cells function, drugs work, and then how can we use that to make our life better, to reduce pain for people, reduce stress, make people better, improve the efficiency of energy production, for example, remove pollution and waste. The important thing is to never stop questioning, is to not stop questioning, not stop researching. You should always be asking why as you as you grow older. Why does that happen? What impact does that behaviour have on this particular individual, and so on. And then there's this whole idea of nature versus nurture. So nature being your DNA and nurture being your experiences. But actually that's oversimplistic. Nature versus nurture is actually, it's a complex interaction between genetic and environmental factors from the moment of conception, which has brought about your uniqueness. So A-level biology at Woodhouse Grove, it is myself, Mrs. Richardson, Dr. Ingham and Mr. Garbutt, and we have Mrs. Dixon is our technician, which I'm sure many of you know, and she's a super qualified engineer, and quite frequently go to her to say, why isn't this practical working, or let's work out some mathematical problems. So she's a really good resource that we have in the department. So these are some emerging and exciting areas of biology that you might want to go into. Synthetic biology, we're now being able to create life, create our own cells, design the genome, and then build cells to do the things that we want to do. So could we design bacteria that can secrete enzymes to break down plastics, for example, or oil in oil spills? Obviously you have the ethics involved with that. Once you've created a new life and you've released it into the wild to reproduce, we can't control it anymore. So there's a bit of ethics to do with genetic engineering and synthetic biology. Immunology, things like allergies, autoimmune conditions, which are becoming more and more common. We're still learning lots and lots of these. And the link between those and our microbiome and all the bacteria that live inside us. Epigenetics is the effect of the environment on the way that our cells use our genes. This is a huge area of research at the moment. Stem cells, can we grow organs from scratch and tissues from scratch in a lab to overcome the problem of transplants? Antibiotics. We did think that the next big pandemic might have been because of a resistant bacteria, although maybe we've still got that to look forward to. Antibiotics, we're struggling to find enough antibiotics and keep up with the fact that bacteria keep becoming resistant to them. And you looked at that a bit at GCSE. Bioinformatics, so we're sequencing huge amounts of DNA from all different species and we're pumping out all this data. So the biologists understand the science behind the data, but they maybe lack some skills in processing it. So we need more mathematicians in biology. The head of molecular biology at Leeds actually came from a physics and maths background, and then he moved into biology to be able to process this data. Epidemiology, which is obviously what's going on now, finding which the next virus will be that will jump into the human population and how we can limit the impact of that. Not managed too well at the moment. Perhaps we need a few more epidemiologists trained to be advising governments on what to do, or the World Health Organization, which might have had a slightly different impact of the coronavirus. Personalised medicine, we're starting to see this with cancer treatment. Before with cancer, if you had a tumour, they'd just give you a chemotherapy drug, and if it worked, brilliant, and if it didn't, your prognosis was poor and it might mean that it became terminal. However, now we're actually sequencing the DNA of cancer cells and realising which cancer drugs work with which cancer cells and giving people the right chemotherapy for that type of cancer, because every cancer cell is completely different. And meaning that we've got much better survival rates from cancer, and we're not wasting NHS money on a treatment that was never going to work in the first place. And then quantum biology, this is a sort of crossover with physics. There's a few different processes in biology, things like respiration, photosynthesis, and enzymes, which actually their action involves the transfer of energy in the form of electrons from one form to another, and that's quantum biology. these are the topics: biological molecules and cells. So these are the first two that you'll start. The bridging work, which I sent out, focused on a bit of a research about biological molecules and learning all the bits of the cell. There's a few more structures than you learnt in year 9. And then the extra bit of the bridging work was about lactose intolerance, because it brings in biological molecules, it brings in absorption, it brings in enzymes, it brings in genetic natural selection. So please be doing that work and contacting me if you want any advice on it. I'll be able to answer these questions until September. I want to collect that work in September. These sessions I wanted to focus on something else to get your teeth into, basically. So we have AS and A-level. We don't actually teach AS, we just teach A-level. So we have 3 papers. One's all about year 1 work, paper 2 is all about year 2 work, and paper 3 is half essay. There's 1 essay that you need to write with 25 marks, and then the other half of it is data interpretation, processing, evaluation questions. So it's sort of a maths and literacy paper for paper 3. So 10% of the marks are maths, 15% are practical on the exam papers, and they might ask you the method for a practical which you did within your lessons, because the other part of the practical assessment is the practical endorsement. We used to have coursework, but what they found was that these schools were just doing the coursework, getting it out of the way, and then not really doing enough practicals. I guess resources were limited, and there wasn't enough equipment to go around. So we're in a really fortunate position where we have more hours on our timetable than most other schools. We get practicals in whenever we can, not just the ones that are on the practical endorsement, but there's 12 required practicals that you'll do over the 2 years. These need to be evidenced in your lab books and will cover a range of skills and techniques. And we need to sign you off to say that you are proficient in those before we can basically award your A-level. Nothing to worry about too much, but if you fall short in your practicals, you'll have to do extra work, basically. We'll make you do them again and again and again until you can show that you are competent in them. But if you speak to some of the year 12 students, they're not too onerous. We guide you through it, and they really enjoy the practical work and having their practical book with all those skills in. This is PowerPoint is on the meeting, so I'll let you go through it in your own time if you want, but these are all the practical skills. Prerequisites. To be accepted onto our A-level biology course, you must have a 5 in science at least, and at least a 5 in your English and maths. However, to be able to make good progress in A-level biology, you really need a 7 in your coordinated science. Obviously, we're going to have to take this year's grades with a pinch of salt, and you'll get a chance to prove yourself in your A-level course. It could be that you really enjoy biology, but not so much the physics and chemistry, so your overall GCSE grade was a little bit lower. You just need to be able to manage your expectations. If you get a set, normally you would get a 7, you go on to do an A-level, and all those people who've got 7, 8s and 9s, the odd 6 in that GCSE would go on to get between A stars and E's in their A-level. If you're getting below a 6 in a subject, you shouldn't be choosing that for A-level. It could be that you didn't concentrate during GCSEs and you mature a lot, but just because you got a 6 at GCSE does not mean a B at A-level. Okay, just be aware of that. We have students on the course at the moment who got 6s and 5s at 6s, sorry, at GCSE. And they're struggling, but they just love the subject. So they're aware they're not going to get the highest grade at A-level. They'll get higher grades in other subjects, but they really want to do it, and they're working really hard. So which subjects will complement the study of biology at A-level? Chemistry. Chemistry can be a bit boring sometimes, but chemistry is the language of biology. you don't have to be a chemist to study A-level biology, but you need to have a good grasp of chemistry to study A-level biology. When you get down to the molecular level, you're dealing with molecules, and it's all the properties and interactions and reactions of molecules will apply to biology. Biology is just the chemistry of life, basically. So be warned, there is some chemistry. Physics, I guess it lends itself well to, although physics is less literacy. There's quite a lot of context, reading, interpretation in biology that you don't get in your physics and maths, and much less in your chemistry. What we find is we have students who do the sciences and they're really good at the interpreting investigation skills bit, because that's the nature of science. We have people who do maths and they're really good at the maths bit. And then we have students who do psychology, politics, English, and they're really good at the literacy side of biology. So being able to interpret what the question is asking for, being able to pick out the key information in any comprehension task that you're given. And they do really well. So really, it's so broad. If you can do all of them, that's brilliant. But there will be different areas that different people are stronger at than others. Higher education courses that biology A-level could lead to: any biological science, zoology, anatomy and physiology, sports physiology, biology itself, which is really broad, molecular biology, genetics, they're all biological sciences. Environmental sciences, so with geology and geography, again, that's going to be a big area when we try and sort of reduce climate change and get a hold on our production of waste and become a bit more sustainable. That's going to be a big emerging area where there's going to be jobs in. Medicine, it obviously lends itself to dentistry, nursing, veterinary science, study of chemistry, psychology, sports sciences, physiotherapy, nutrition and education. It all lends itself to. But actually, even if you're not going to go into the sciences, it's really great facilitating subject. If you have an A-level in biology, that immediately tells someone that you are good at literacy, that you can cope with interpreting questions and problem solving, that you have maths and investigation skills, are aware of ethics, and they can apply to so many different subjects. So that's why biology is a perfect subject for you to study. Oh, just one more slide about what we as a department would expect from you. You're not going to make much progress if you're not in lessons. You need to be in lessons. You need to meet homework deadlines, because the way that we plan our marking, if you hand it in, you get marked, you get feedback, that's going to help you progress. If you miss the deadline and hand it in at a later date, you might not get that feedback that you need. Obviously, sometimes you're going to be like, I've had this on, or this has happened to me, I need an extension, but don't make it a frequent thing. You need to go and consolidate your work done in your class in your own time. I expect you to be in the lesson and to be able to have understood everything from the previous lesson. Okay. Ask specific questions of your teacher. We generally have much smaller classes at A-level than GCSE. It's quite a nice, fun, relaxed atmosphere most of the time, and really supportive. So take advantage of that and build relationships with your peers and the teacher and ask questions. Come to drop-in, where you can just come and sit and do some work with some teacher support. Let us help you. That's what we're there for. Come in on time to lessons and arrive fully equipped. What you should expect from us is that we will deliver engaging lessons, and we will make them interesting, and make you think, wow, or I didn't know that. And you go away, hopefully, wanting to learn more about the subject. You need to prepare students for your exams. Oh, sorry, we need to prepare students for exams. We need to be able to know exactly what you have to know. We need to skill you up to be able to do well in your exams. We should have excellent subject knowledge. The teachers and I are always reading and expanding our knowledge and having discussions. We love the subject and we like to test our knowledge and gain new knowledge, and that's what you should have from a subject teacher. Provide individual feedback when possible to enable progression, and we should communicate with you, parents and the sixth form team in terms of your progress, so you don't fall through the net if you're having a bit of a tough time. Right, is there any questions on what to expect from A-level biology? Right, I am going to get up today's lesson. So this is on Firefly. It's under year 11 if you have access to Firefly. I'm sorry, I don't think the external students do, but I can copy and paste this as well. So what is zoonosis? Has anyone heard of the term? Should I pick on someone? Tom Vernon?
Yes, miss.
Have you heard of the term zoonosis?
No. Does it have anything to do with animals?
It does. Why do you say that?
Because zoo.
Yeah, exactly. So zoology is the study of animals. So zoo is animals. Cis generally means infection, bronchitis, arthritis. It means infection or inflammation. So it's infection to do with animals. Okay, well done for having a go at that, Tom. So I hope we can all still see the screen. So what is zoonosis? Each species that exists, including bacteria, which are only single-celled species, and the cells are very different from ours, have evolved alongside viruses since life began to evolve. You could either go two routes. They either went down the sort of complex cellular route, where you display all the life processes and they can reproduce, they respire, sensitive, they excrete, they take in nutrition, or they went down the simple route of viruses. Now, all viruses are is they're basically a little genetic code inside a little protein cage, which is covered in fat and some more special spiky proteins on the outside. Those proteins that are on the outside basically act as keys that allow them to get into a specific cell of a specific species. Okay, and so these have very intimate and ancient relationships with their host cells, the cells that they have the key to get into. A virus cannot reproduce itself. It doesn't have all the things in the cell which enable it to reproduce, which is why it's so small. It doesn't have to have any of the other cell machinery in it. So they invade a cell and they basically just inject their genetic code, their instructions to make new viruses. And then the cell, unwittingly, doesn't know what it's doing, starts reading that code and using the ribosomes and all the different enzymes that the cell has to make new virus particles. And then they fill the cell and they burst out of the cell and destroy the cell. And then they move each one of those thousands of viruses which has been invaded, goes and finds a new cell to invade, gets reproduced 1,000 times, and so on. And after so many cells are destroyed, you're going to start to have some symptoms. So they have ways to affect the host and also enable transmission. They want to get to a new host, especially if that virus affects the cell and so much that, or the organism, that's going to kill it. It's no good killing your host, because then what are you going to do? So if they are quite dangerous, they need to, if they're damaging the host, they need to be able to get to a new host, okay? Because if they kill the host and they're inside, they're gone as well. So things like sneezing and coughing is a good way to propel those virus particles to reach a new host. So when the viral genetic material is being copied by the cell, sometimes there's little mistakes that's made in the code. You know, if you're copying from the board, say, and you're looking up and down, you're writing, you might make the odd mistake in your notes. That's what happens when virus genetic code is being copied. Little mistakes happen. This is more common in viruses that have a type of genetic material called RNA, which is like DNA's cousin almost. It's much simpler, and because it's simpler, mistakes happen more frequently. Sometimes these mistakes actually have no impact at all. Sometimes they might be detrimental to the virus, but occasionally a mistake that's made when copying that code can actually result in the production of a new type of key, those little protein spikes on the outside of a virus, a key which will allow it to get into a new cell, maybe in a new species. And again, that key is irrelevant. The fact that that virus has a new key is irrelevant if that virus does not get into the species where the key fits. So if circumstances allow that virus inside that host animal to come into contact with another species, which it has the key for now, then it will be able to infect that new species. But those circumstances don't always happen. When it does happen, it's called a spillover event, spilled over from one organism to another. Now, sometimes that spillover event is just a one-off. It infects that new individual. That individual will either get better or die, and that's it. But sometimes that virus is able to then be transmitted between individuals of this new host species. And if that's the case, none of the new host species have any immunity to it, and it spreads like wildfire. And this is what is called a pandemic. And we all know how they unfold. I'm just going to play a clip. Please shout out if you can't hear the sound.
Zoonoses refers to diseases that are spread from animals to humans. It's possible for humans to catch diseases from a range of invertebrate and vertebrate animals. The term vertebrate refers to animals that have a backbone, like cows, pigs, bats, and birds. The term invertebrate refers to animals that do not have a backbone, like ticks, mosquitoes, and sandflies. It is also possible for humans to transmit diseases to animals, and in this context, the zoonosis is termed a reverse zoonosis. The risk of infection with zoonosis increases as the human population continues to grow and people live in greater proximity to animals. While the term zoonosis applies to all diseases that are spread from animals to humans, the term vector-borne diseases applies only to those spread by an intermediate carrier. An example of an intermediate carrier is a mosquito. An example of a vector-borne disease is malaria. The mosquito bites an infected animal, such as a cow, shown here, and becomes a carrier of the malaria pathogen. At this point, the mosquito is referred to as a vector. When a mosquito bites, it releases a chemical into the circulation to stop the blood clotting. During this process, it could also release one or more of the malaria parasites it has been carrying into the circulatory system of the human it has bitten. If it does this, the person becomes infected. Once in the circulation of the infected human, the malaria parasite travels to the liver and multiplies. After multiplying, the malaria circulates again in the blood. It is possible to take action to prevent infection with the malaria parasite. For example, you can use insect repellent and you can use insect nets while sleeping. You can also avoid travelling to countries where malaria is a known risk. These are simple prevention methods that avoid contact with the vector.
Get more from the Open University. Check out.
Okay, so that was an example of malaria, which hopefully we've all heard of. It kills hundreds of thousands of people every year. We can take anti-malarial tablets if we want to go to one of the countries where malaria is common. It's also common in warm places where there's stagnant water, because that's where the mosquito breed. So I'm not sure if anyone's been anywhere exotic where they've taken anti-malarial drugs. Put in the chat if you have. So that's just one example. So zoonosis is a disease which can be passed from animals to humans. That was a parasitic protist, that the Plasmodium, which causes malaria. It's not a virus, that one. So we've got one more clip, and then I'm going to set you off on some tasks. So how do viruses jump? So this is viruses.
At a Maryland county fair in 2017, the prize pigs were not looking their best. Farmers reported feverish hogs with inflamed eyes and running snouts. But while fair officials worried about the pigs, the Maryland Department of Health was concerned about a group of sick fairgoers. Some had pet the pigs, while others had merely been near their barns. But soon, 40 of these attendees would be diagnosed with swine flu. More often than not, sick animals don't infect humans, but when they do, these cross-species infections, or viral host jumps, have the potential to produce deadly epidemics. So how can pathogens from one species infect another? And what makes host jumps so dangerous? Viruses are a type of organic parasite infecting nearly all forms of life. To survive and reproduce, they must move through 3 stages: contact with a susceptible host, infection and replication, and transmission to other individuals. As an example, let's look at human influenza. First, the flu virus encounters a new host and makes its way into their respiratory tract. This isn't so difficult, but to survive in this new body, the virus must mount a successful infection before it's caught and broken down by an immune response. To accomplish this task, viruses have evolved specific interactions with their host species. Human flu viruses are covered in proteins adapted to bind with matching receptors on human respiratory cells. Once inside a cell, the virus employs additional adaptations to hijack the host cell's reproductive machinery and replicate its own genetic material. Now, the virus only needs to suppress or evade the host's immune system long enough to replicate to sufficient levels and infect more cells. At this point, the flu can be passed on to its next victim via any transmission of infected bodily fluid. However, this simple sneeze also brings the virus in contact with pets, plants, or even your lunch. Viruses are constantly encountering new species and attempting to infect them. More often than not, this ends in failure. In most cases, the genetic dissimilarity between the two hosts is too great. For a virus adapted to infect humans, a lettuce cell would be a foreign and inhospitable landscape. But there are a staggering number of viruses circulating in the environment, all with the potential to encounter new hosts. And because viruses rapidly reproduce by the millions, they can quickly develop random mutations. Most mutations will have no effect, or even prove detrimental, but a small proportion may enable the pathogen to better infect a new species. The odds of winning this destructive genetic lottery increase over time, or if the new species is closely related to the virus's usual host. For a virus adapted to another mammal, infecting a human might just take a few lucky mutations. And a virus adapted to chimpanzees, one of our closest genetic relatives, might barely require any changes at all. It takes more than time and genetic similarity for a host jump to be successful. Some viruses come equipped to easily infect a new host's cells, but are then unable to evade an immune response. Others might have a difficult time transmitting to new hosts. For example, they might make the host's blood contagious, but not their saliva. However, once a host jump reaches the transmission stage, the virus becomes much more dangerous. Now gestating within 2 hosts, the pathogen has twice the odds of mutating into a more successful virus. And each new host increases the potential for a full-blown epidemic. Virologists are constantly looking for mutations that might make viruses such as influenza more likely to jump. However, predicting the next potential epidemic is a major challenge. There's a huge diversity of viruses that we're only just beginning to uncover. Researchers are tirelessly studying the biology of these pathogens, And by monitoring populations to quickly identify new outbreaks, they can develop vaccines and containment protocols to stop these deadly diseases. Okay, how exactly do vaccines work? We break down why vaccines are safe and necessary in these.
Okay, over this series of lessons, we will be looking at vaccines, herd immunity, how you come up with a vaccine and go through proper trialing, drug trialing processes, and how we might actually eradicate a virus. So they've called it host jump in that video. We'll call it a spillover event, is when a virus passes from a reservoir population where it has naturally existed to a new host population. It may or may not be able to be transmitted through the host population. If it can, it will spread widely. So in terms of the SARS-CoV-2 virus, being airborne is a really effective way to transmit. Okay, if you have to be exchange blood, it's much harder for you to jump between individuals. But we've seen with other viruses, hopefully we'll do some research, that is obviously still possible. And we still get epidemics because of it. But being airborne, you know, we have to breathe. I don't know if you've seen some of the research that the SARS-CoV-2 virus, which causes the the COVID-19 disease, can exist if you've got it on your hands because you've sneezed or coughed and you've put it on a worktop, it can exist there for sort of 10-12 hours. If it's stainless steel and not very porous, it can exist there for about 48 hours. And then so 48 hours, any time within that period, if someone comes and then touches it and then touches their eyes or, you know, then they will become infected. So it is really good at spreading, is this new virus that we've got. So I want you to just do a little bit of research. I'm not sure about you, but the first thing that I did when we got locked down is I ordered this book. This is called Spillover, and this author, David Quammen, I read an article by him years ago, like 2004 or something, about him he's like a virus hunter, hunting for the next virus, which is the next virus that's going to jump from animals. And I was really interested in it, and I've kind of done bits of reading over time. But as soon as we went to lockdown, I bought this, and I did show some people, like Dr. Ingham. She was like, "What are you doing? You know, it'll just stress you out and cause you anxiety." But for me, I've always found the best way to deal with anything that causes me confusion is to find out more about it, understand it properly, and then it won't cause you anxiety, because I find a lot of anxiety is mixed up with being ignorant of something and being confused and not knowing what's going on. So the best way forward is to educate ourselves on the topic. So I want you to do a couple of tasks. There is an excerpt, 6 pages, which might seem a lot to read, on the Teams channel, if you can download it. I must apologise. I had my book out with a pen last night. It was all pristine, and my 2-year-old has scrawled on some of the pages, so you'll see some random markings on there. So that is your first task, to read that, and I want to hear what your thoughts are on it. Task 2, I want you to have a little think about different viruses. In those 2 clips the first one was about malaria, that's not a virus. So they talked about swine flu in that TED talk. I'm sure we've studied something, and we definitely have studied something in year 11 about a virus that jumped from animals at the end of your reproduction topic. And there'll be some other viruses. You could use SARS-CoV-2 as one as well. So which animal did it originate from? Which country did it appear to have made the jump, the spillover event occurred in? And what are the fatalities? And then finally, what I want you to do is read this article. Some of the ideas were alluded to in those clips. So if you click on this link. Human impact on wildlife to blame for spread of viruses. So people actually think it's what we are doing is increasing the frequency and likelihood of these spillover events. And also more things that we're doing are meaning that it's more easily transmitted within the population, and pandemic is more likely. So that's the article. It's not too short, but again, I read this very early on in the pandemic. When did it get published? Yeah, it's not a date there. That's strange. Oh, Wednesday, the 8th of April. So have a little look at that. And then can you just jot down some answers? And I'm going to pick on people to share your ideas. So these questions say suggest, suggest, suggest. So it's you reading it and you trying to make a sensible, I come up with a sensible idea. So suggest what human practices are making spillover events more frequent. Suggest ways that human practices have encouraged a high transmission through the population. How is it able to spread? And also suggest what we could do. So off the back of this, what should the governments now be putting in place if there is a next spillover event and it does transmit, which is highly likely. It could be 10 years, it could be 50 years, who knows, but it will happen again. What can we do to stop it being such a global catastrophe as it has been at the moment? And we'll share your research next lesson. Okay. Has anybody got any questions? No? If you're a bit shy and don't want to ask your questions out loud in front of everyone, or you just want to send me a message, that is fine. Please do that. Have a real think and get some reading done. If actually finding out about the virus or viruses is actually causing you stress and anxiety, please don't do it. Just let me know. But I wanted you to do something different to your bridging work because you've got a lot of time on your hands. And knowledge is power, okay? And knowledge makes you more better equipped to cope with the things that are ahead, in my opinion. Okay, shall I bid you a farewell? Bye then.
Cheers, miss.
Bye.
Thank you, miss.
Thanks, miss.
Thanks, miss.
Bye. Nice to see all your friends. Bye and hear you.